My initial plan, I have Mary's resume on my cell phone. I just opened it up. My initial plan was to just thumb through it and read a few highlights to you, but the damn thing's like 100 pages long, and I can only see about a sentence at a time. So I'm going to have to try and wing it. So I'm actually being sincere when I say it's a genuine honor for me to welcome Mary Carrington to her first Cold Spring Harbor Retroviruses meeting. Hopefully we'll get you to come back to future meetings as a regular attendee. Of the people that I know who work in the HIV and AIDS field but don't regularly attend this meeting, Mary Carrington is the one that most deserves to be considered an honorary member of that society of people who attend and appreciate the Cold Spring Harbor meeting. And I'm saying that not based on her scientific achievements, which are considerable, but based on the fact that she's probably one of the most collegial and collaborative people in the HIV/AIDS field. I've never heard anybody say anything bad about her, actually. So let's not start now. Those of you who know Mary will not be surprised to hear that she told me that I didn't have to list any of her awards and many achievements tonight. So I promised her I wouldn't do that, but I lied. But as a compromise, I'll keep it short and sweet, and I just have sort of 3 points I want to touch on. So the first is just the basic facts. So Mary did her PhD at Iowa State University in Ames. She postdoced at Duke and at UNC Chapel Hill. And then in 1989, she became a principal scientist at NCI Frederick, where I think she's been ever since, and she's also currently the director of the Basic Sciences program at NCI/SAIC. I'm not sure what that means. Oh, Lino. You need to update your CV. She's also a visiting professor at Harvard Medical School. The second point, then, that I wanted to get to, if I can extract one thing from her CV that I'd like to point out, that is in addition to numerous invitations to speak in numerous countries, as far as I can tell, there's also an unusually high number of visiting lectureships and visiting professorships, and I think that also speaks to the fact that she's an extremely collegial person and very helpful collaborator to a lot of people in the field. So if I can highlight that one thing, I consider that a major achievement. The third thing, and it's sort of my personal view on Mary's career, which I followed since about 1995 when I was a graduate student in John's lab, and he took his turn in our lab journal club rotation because he was one of the rare PIs that actually did that once in a while. And he presented one of the papers from the genetics room down at NCI Frederick, and so I've been following Mary's work ever since. And what's very impressive to me is her work in particular on the immunogenetics of HIV infection in human populations, right? Because there is no animal model that allows you to do simple classical genetics in this when you're asking these sorts of questions. So it really relies heavily on population genetics and statistical genetics. And if that's not, that's a considerable feat in and of itself. And then to layer on top of that the fact that she has chosen to focus a lot on the human MHC loci. And for those of you who aren't aware of it, the genetics of the MHC are basically insane. And she must be a glutton for punishment because on top of that, she has layered also looking at the genetics of the cure loci, which if MHC is insane, I think the cure loci are ludicrous. So very complicated stuff and probably one of the best people working on it in the field. So Mary, if you'll come up and help us understand the complexity.
Well, thanks very much, Welken and John, for the invitation to be here tonight. I'm really delighted to be here, and I will be back. And I'd really like to thank the audience for your willingness to spend the next 40 minutes or so listening to immunogenetics. What I'd like to do is start with just a tiny bit of historical perspective and really concentrating on two people who I sort of flatter myself to think of them as part of my scientific pedigree. And the first one is Peter Gorer, He's the person who discovered the Major Histocompatibility Complex in mouse, which is called H2. He did that along with George Snell, and in 1980, George Snell won a Nobel Prize for that work, and by that time, Peter Gorer was no longer living, and certainly he would have shared in that prize had he been living. He also really pioneered the field of transplant immunology, and he was a contemporary of Peter Medawar, who won a Nobel Prize for his work in transplant immunology in 1960, and I think probably Gorer should have shared in that prize as well, because he was doing this along the same paths as Medawar. But Gorer was not a self-promoter. He wasn't the easiest person, and he basically didn't win any prizes. He was not the kind of person to win any prizes because he was a little bit acerbic, and he loved to drink scotch and drank way too much of it. He was a copious smoker. And you can see he was known for speaking to people with a cigarette hanging out of his mouth. And in general, he didn't do much to present himself for any kind of prize. But the guy was absolutely brilliant. And he laid the foundation for the next 55 years of the work in the MHC. And I think he's probably still not appreciated for as much as he's done. Now his protege was Bernard Amos, and Bernard was one of my mentors at Duke University. Bernard was also a really accomplished scientist, and you can kind of read through these here. He started out in H2, but then really moved to the human MHC, which houses the HLA class 1 and class 2 genes. And the thing I'd have to say about Bernard is that he was extremely unselfish in the work that he did. Although being a very bright man, he really organized the field, and one of the big steps in that regard was organizing the first International Histocompatibility Workshop, which, as far as I can tell, is really the first scientific consortium to be organized. And this is different than the ones that we deal with today, where we get on the phone call and we all talk about our specific disease and do meta-analysis on data that everybody's generated. In this one, he had the 10 to 15 people throughout the world who were working on HLA and trying to understand the serological basis for that, so that individuals who needed a transplant we'd be able to identify appropriate donors. Bernard had these scientists, along with their key technicians, come to his lab at Duke, and he forced them to pass their reagents around so that the other scientists could test those reagents of their colleagues. And this was not an easy thing to do. I mean, can you imagine if you brought together people who think they have a new virus, and then you bring them into a room, and then you pass those viruses around and have the group determine whether you actually found a new a virus in humans, or whether it's a contaminating mouse virus that popped into some tissue culture. But this is what he did, and people were wrong about some of their reagents, and everybody got over it and moved on. This was not a job that one person could do, because this region is so complex. And I thought I had only half an hour to speak, and unfortunately, Welkin informed me that I had a little bit more, so I'm just going to tell you one story. And Bernard was reportedly a very precocious child, and developed into quite an eccentric adult. I think that trajectory is not uncommon among scientists. I think there are probably a few in this in this audience that I know that have taken that route. And, I'll just tell you one short story about Bernard, and then I promise I'll get to some science. The HLA was thought to possibly be involved in mate choice, and part of that was because there was thought to be a greater number of heterozygotes for HLA class 1 and class 2 than expected, given the number of alleles that had been identified. And so it was thought that this might have been done, might be due to some kind of selection pressure. So you want heterozygosity, you want to find a mate who's not so much like you genetically, because that's inbreeding. And so the idea here, and furthermore, being heterozygous would reportedly give heterozygote advantage, and there is support for heterozygote advantage in multiple diseases, including HIV. And so Bernard, this idea that HLA was involved in mate choice was really interesting to him. And the idea was that this was due to smell. And so when he moved, when he retired, he moved from one building into the building where my lab was. And after all the dust settled, this box remained in his office, this big box remained in his office until the time I left. And what was in that box was a whole lot of men's underwear. And, and Bernard had designed this experiment where he was going to have a bunch of men, okay, not bunches, not scientific. He was going to have a cohort of men wear this underwear for 2 or 3 weeks, and then this is the truth. And then he'd have a cohort of women that he organized that would smell the underwear and decide which was the most attractive. And I think least offensive. And so the experiment, so he was able to get enough men who were willing to do that, and he could pay them to do that, and some he probably didn't have to pay, and so, but he had a real problem coming up with enough money to identify the cohort of women who were willing. So the underwear just sits in Bernard's. And I've often wondered how people who wear perfume or men's cologne, whether if we, that sort of screws up this whole idea about mate choice. And I wondered whether maybe people who wear perfume might actually be more inbred than those who don't. Anyway. Okay. So anyway, just an idea. So the birth of the MHC was really within transplantation immunology. That's how this came about. But over time, we know that the variation at these loci is not due to, it's not there to keep us from accepting grafts from other people. And it soon became apparent that the real purpose of these genes is to protect us from infectious agents and cancer. So since that time that I've been talking about it, there's really been an explosion. And Zinkernagel and Doherty won a Nobel Prize for defining the true function of these HLA molecules in antigen presentation. And so now we have found as a community that these genes within the MHC associate with a huge number of diseases, and I'm only putting a few of those there. They're hugely polymorphic genes. All of these diseases pretty much associate with variants nearby the classical class 2 genes or class 1 genes. And this is really the most rich 4 megabase region of the human genome in terms of its association with diseases. We've known that since before GWAS, genome-wide associations, came about. We knew many of these associations prior to that. But what GWAS did is it told us how important this region is relative to the rest of the genome. And historically, the basis for these associations has been thought to be the role of class 1 in antigen presentations to the T cell receptors. So this region encircled in green is the part of the peptide binding groove. You see the peptide sitting right there. That's the part of the peptide binding groove where the polymorphisms determine which T cell receptor will recognize that molecule and initiate an immune response. And since that time, and much more recently, another part of the class 1 molecule, and I'll be talking about class 1 in particular today, is also polymorphic and recognized by another set of polymorphic receptors called the killer cell immunoglobulin-like receptors. And those are expressed on natural killer cells, and they regulate the activity of natural killer cells in a subset of CD8 positive cells. And the identification of class 1 as a ligand for KIR made it clear that these molecules are not only involved in the acquired immune response, but also the innate immune response, and that could account for some of their disease associations. So what I'm going to do is kind of walk through 5 characteristics of the class 1 molecules that we think are having effects on disease and that need to be taken into account when we think of disease. And this is all in the context of HIV, where we have huge cohorts large enough to be able to interrogate these genes for their characteristics that are having effects. And here we're looking at single allelic effects. As I mentioned, these individual alleles will have an effect on what CTL is going to respond to a given antigen. And these alleles in people of European descent are protective, the ones below the bar. These are associate with susceptibility to different levels, as you can with different levels of significance. And the thing that's pretty amazing, okay, so here's B57, and I'll come back to that later. B57 is really the most renowned allele in terms of HIV. It confers protection, but actually most people who have B57 progress to disease if they're not on therapy. Now, one thing about this slide that I think is important is that if you look in people of African descent, there's not a single 4-digit allele that matches those that associate with HIV in people of European descent. And that really speaks to the incredible diversity of these loci and the difference across ethnic groups in terms of the frequencies of these alleles, and in some cases whether the allele is even present or not. And a lot of this work really has been performed by Gao Xiaozhen, who's from China originally and now working in the lab, and Arman Basharova, who is from Kazakhstan. These two have really pushed this aspect forward in the lab. Now, a few years ago, and this is not work from my lab, but rather from Jacques Neefjes in the Netherlands, Jacques' group showed that HLA class 1, or MHC class 1 in the mouse, is upregulated on this tumor cell line after radiation treatment in vitro. And that's just shown here. H2K of B is a class 1 molecule. It's upregulated upon irradiation. And then they went on to show that in situ irradiation of the tumor, okay, so they inject the tumor and then they irradiate it, followed by injection of tumor-specific CTLs, led to arrest of the growth of the tumor, okay? And that's just shown here. So they give tumor here at time 0, At about day 9, they irradiate the tumor in situ, and then a few days later give the CTL specific for that tumor, and they arrest the growth. And any one of those without the others has no effect on tumor cell growth. So what this showed was that enhanced class 1 expression results in increased efficacy of CTL in vivo. And that was really a nice prelude to some work that Richard Apps, who comes from Cambridge, England, and is a postdoc in my lab, that the work that Richard did where he showed that HLA-C allotypes are expressed at different levels on the cell surface, and individuals who have high expression are protected against HIV disease, relatively speaking. So high expression of HLA-C, this is a classical class 1 gene in humans, high expression of HLA-C associates with protection against HIV, but actually associates with susceptibility to an inflammatory bowel disease, Crohn's disease. Low expression protects against Crohn's. Low expression also in unrelated hematopoietic cell transplantation, low expression of a mismatched HLA-C allele in the patient protects that patient against graft versus host disease and non-relapse mortality. So expression levels of these molecules, we think, are having an effect on disease. They're modifying the effects of individual allelic effects. And this is probably the tip of the iceberg. I imagine that expression levels are having an effect on a lot of diseases. This is Jonathan Carlson. He works at Microsoft. He's actually American. He lives in Seattle. And we generally try to generate some functional data to support the genetic data. And Jonathan was working with a very large data set that came from Philip Goulder at Oxford and Tumbi Nungu at the University of KwaZulu-Natal, where they had collected material from over 1,000 South African individuals, and they looked at CTL responses in each one of those individuals, looking at peptides across the HIV proteome. So they knew for each one of those people which peptides they would respond to. And Jonathan took this huge data set and asked whether you would have better CTL responses, greater CTL responses if you had higher expression, which would support the genetic data. And of course I'm telling you this because that's the case. This dot right here, so each one of these dots represents people with a different HLA-C type. C1402 is our highest expression allele. Individuals with C1402 are much more likely to respond to their 6 C14 restricted epitopes than are individuals who have C0702, one of our lowest expression HLA-C alleles. So really nice functional data to support that genetic data. The higher the expression of HLA-C, the more likely individuals with that C allele are going to respond to their given set of peptides. Now, a third aspect of these molecules is that we think that probably they are interacting, we think they are interacting with variation elsewhere in the genome. And these types of things need to be pulled out. If you think about ApoBec or TRIM5-alpha, we don't pick these up in a GWAS, but whether there's actually a variant within or near those genes, perhaps having an effect on their expression, that is interacting with an unlinked variant, that would never be picked up in a GWAS. And this is another effect that was not picked up in a GWAS. And I'll just walk you through this. This is data from Smita Kulkarni in the lab. She's from Bombay, India. Smita had shown that HLA-C alleles in the 3' UTR of some HLA-C alleles, there's no binding site for this microRNA. But it's a polymorphic region, and some HLA-C alleles do have an appropriate seed region for the microRNA. And those alleles that do bind the microRNA, and there's downregulation of HLA-C through miR-148A. Okay? And that is one of the one mechanism for differential expression. It doesn't explain the entire expression array. Now, what Smita found was that when she went over to chromosome 7 in the microRNA gene, she identified an insertion-deletion polymorphism that associates with the microRNA expression level. So having the insertion at this position associates with low-level expression of the microRNA. That has no effect on this one because there's no binding site, but it does downregulate to some extent alleles that do have the microRNA binding site. Now, having a deletion at that position, on the other hand, results in high concentration of the microRNA and greater downregulation of those alleles that have the seed region for that microRNA. We get low expression, poor control of HIV, but protection against Crohn's disease and graft versus host disease. It doesn't matter if we have high concentration for those alleles that do not have the binding site. They don't recognize it and they're not affected by it. The point here is that this variant within this microRNA gene has a significant effect on HIV outcome when we stratify our patients by whether they have the microRNA, HLA-C alleles with the microRNA binding site or not. Among individuals that have HLA-C alleles with the microRNA binding site, this variant, the insertion-deletion polymorphism, has a significant effect on HIV control. But among the group of individuals who have two C alleles that do not bind the microRNA, there is no effect whatsoever of this locus. And I think that's something that we really need to keep in mind consistently when we look at GWAS results. GWAS is looking at a single variant at a time. It does not consider multiple variants unless somebody does something in particular, and I'm not aware of it. Now, we went on to have a look at HLA-A expression, and much to our dismay, A expression in our patients associates with poor control. And we were really upset about this because we thought higher A expression, greater CTL activity, and more protection, but that's not the case. And we have good power for that. And we see it in people of European descent. We see it in people of African descent. And these are the two people who have been working on that. Veron Ramsaran, who's from Durban, South Africa, and Danilo Augusto, who's from Curitiba in Brazil. They've been trying to figure out and coming up with models to explain why would high A expression result in poor outcome when high C expression is so protective. And one idea has come from another postdoc in the lab, Nico Vance, who's from Brittany in France. And Nico's been looking at these expression quantitative trait loci. Now, as it turns out, variation within the MHC and near to the HLA genes within the MHC actually regulate expression levels of unlinked genes. And it's been estimated that 40% of the eQTLs within the human genome land within the MHC. So genes in that region are controlling somehow, and we don't know how, the expression levels of genes that are unlinked to them. And we think that that could influence disease pathogenesis, and it could explain the mechanisms for why HLA associates with so many different disease types. So I'm just going to walk you through one example that I think is sort of clear-cut. This is a SNP that is located close to HLA-A. This SNP in our GWAS of nasopharyngeal carcinoma and in another large GWAS of NPC is the top hit in determining NPC risk. Now, we've known way before GWAS that A1101 associates with protection against NPC. This SNP marks A1101. So it really wasn't very surprising that that SNP associated with NPC risk. As it turns out, based on this, this study from Westra et al., we weren't involved in that, this SNP associates very strongly, 10 to the minus 98, it's just outrageously strongly, with this gene that encodes a tumor suppressor, TSSC4 on chromosome 11. So that really gave us a new mechanism to consider why A11, we always figured, oh, A1101 is presenting some EBV peptide that results in protection against NPC, but it's entirely possible that A1101, for some reason, is actually affecting the level of a tumor suppressor gene, and that may be, at least in part, the association between A1101 with NPC. Now, Nico also identified this variant, which actually marks HLA-A expression levels, but not very well. It is significant, but it's weakly significant. And this variant in another database called SCAN database associates with expression levels with HAV-CR2. And that's on chromosome 5, and it encodes a molecule called TIM3. So TIM3 is an inhibitory receptor that's expressed on exhausted CD4 and CD8 cells in chronic infections. It also is defective in a couple of autoimmune diseases. It binds a molecule called galectin-9, and that leads to Th1 cell death and downregulation of T cell responses. It leads to peripheral tolerance. So the question then was whether high HLA-A expression levels might lead to higher TIM3 expression levels and greater exhaustion of T cell responses in HIV-infected patients. And that might explain why high A expression actually associates with poor outcome of HIV. Veron and Danilo have looked at, tested for a correlation of TIM3 cell surface expression levels by HLA-A mRNA expression levels. And they see a positive correlation in CD4 stimulated T cells and CD8 stimulated T cells. And this has led us to the model. So the higher HLA-A expression levels on, these are from normal individuals stimulated in vitro. The higher the level of HLA-A expression, the greater the TIM3 cell surface expression after stimulation. And so the model that we're working on presently, one of the models, and I won't be able to talk about others today, but one of those is that poor HIV control among individuals with high A expression may result in part from higher TIM3 expression and greater exhaustion of T cell responses in HIV-infected people. And we're looking at other exhaustion markers as well, such as PD-1 and LAG-3. Okay. So the role of HLA class 1 in regulating expression levels of unlinked genes, genes that we never had any idea were in any way correlated with HLA functionally whatsoever. And I think that is an area that we really need to probe closely, especially because, as it turns out, there are actually quite a number of oncogenes and tumor suppressor genes that are associating with that. Okay, so the final thing I want to tell you about is the role of HLA class 1 allotypes serving as ligand for innate immune receptors. And one of those set of receptors are the killer cell immunoglobulin-like receptors There's another set, and I won't have time to talk about that, called the Lillers, but today I'm going to tell you a little bit about KIR. And this work, the KIR work in the lab, is really run by Pat Martin. She's from Kingston, Jamaica. And what's been known is that these inhibitory receptors, inhibitory KIR, which are expressed on natural killer cells, bind HLA class 1, specific allotypes of class 1. And when they do that, and class 1 looks normal, normal level, normal self-peptides in the groove, they send a signal, it sends a signal to the NK cell not to kill that cell because it looks normal. It's perfectly normal. And that inhibition will be overridden by any kind of activating receptor recognizing its ligand. So this is a simplification, but in general, that's the picture. And upon HIV infection, NEF downregulates HLA-A and B, and whatever is left may be altered. It may have stress peptides sitting in the groove or HIV peptides sitting in the groove, and it doesn't look normal anymore. And that inhibitory receptor does not recognize it, and an activating receptor can now send a signal to kill that cell because it's aberrant. And so I'm going to come back to this. Now, this is David Goldstein. He's not in my lab. He's at Duke University, and he's never been in my lab, and he's an American. And the reason I bring this up is because I've been questioning why I never seem to have any Americans in my lab. And I don't have a single one right now. I rarely have one. And it's really not that I don't like them. I married one, and forgot, and I gave birth to 2 of them. So, I do work with a lot of Americans, and I was thinking about this earlier today. I think I have someone from every continent of the world in my lab, one person from every continent except North America and I guess Antarctica. Anyway. No, I don't think so. And then I wouldn't get my husband, But I always thought the reason that I sort of was attracted to my husband was because of morality and intelligence, but I've begun to wonder, and I don't think it's smell. But anyway, what we, he would not like me saying that. And we're not matched at HLA. Anyway, so with David, we selected about 100 elite controllers, and all of these people have B57. What we were asking is there any locus genome-wide that is modifying that B57 protection that I mentioned earlier? So we have about 100 elite controllers, and we selected the most elite of the elite B57 controllers. And then we picked about 100, we tried to get 100, they didn't all get sequencing, but 100 of B57 people that were the worst of the non-controllers. So we really tried to get the extreme ends of any samples that we could get. And we sent those down to David's lab, and he did full genome sequencing. And only one single variant in the KIR3DL1 gene, this is an inhibitory receptor, one of the KIRs expressed on NK cells. Only a single variant, it's a nonsynonymous variant that switches an isoleucine at position 47 to valine. That was the only one that came up genome-wide. And when you do genome-wide on whole genome sequencing, you have to start out by choosing candidates. And we chose about 60,000 candidates for quite a number of reasons. And this was the only one that reach genome-wide significant. I think these two are pretty much an LD with that one, and I don't know what that's all about, but it's not very significant, and it's nowhere near genome-wide significant. So we've gone on to look at a larger number of B57 people, and to get those larger numbers, we loosened the definition to less than 2,000 and greater than 10,000 copies of the virus per ml of plasma. But this holds up nicely. Individuals who have 2 copies of isoleucine at position 47 are more likely to be a non-controller. Not a lot more likely, but they're more likely to be a non-controller. And those that have at least 1 valine at that position are more likely to be controllers. And these differences reach significance. So we think that the one, given the power that we have, that the one locus that is modifying B57 protection is actually KIR3DL1. We have no idea why that's the case. This is a crystal structure from Jamie Rossjohn's Nature paper a couple of years ago, where this is the KIR3DL1. This is KIR3DL1 right here. This is actually B57. So this is the class 1 molecule. This is beta-2 microglobulin, and there's the peptide sitting in the group. And if you look at where position 47 is, it's pretty far away from the class 1 molecule, and hard to imagine that it's having any real effect on the interaction between these two. But I guess it could affect the structure, which could affect their interaction. Position 2, 47, and 54 are in near complete linkage disequilibrium with one another. If you know what's at one, you likely know what's at the other across the alleles of 3DL1. That's quite a polymorphic gene. So if you have valine at 2, you have valine at 47 and leucine at 54. The alternative is methionine, isoleucine, isoleucine. And when we went back, okay, so position 2 would be right around here. But the structure was actually cut off at amino acid number 6, so 2's not actually in the structure that Jamie published. But what an older paper from Peter Parham's group showed that if you start switching these, a single amino acid change in the allele that has VVL, remember the valine was protective, you can affect the level of binding of an A24-Neph tetramer. And in fact, having a methionine at position 2 actually increased the binding between that tetramer and that KIR3DL1 molecule. So this could have something to do with it. This may be why that having stronger binding is leading to greater inhibition and perhaps inhibiting the NK cell activity to a greater extent. And that's a bit of hand waving, but that's where we are on that. The only thing we feel sure about is that 3DL1 is actually modifying a B57 effect. Okay, so what I've tried to do is just give you an idea about some of the characteristics of the class 1 locus in terms of their effects on diseases and things that we really need to think about beyond just single allelic effects and the type of peptide that an allele is presenting. Certainly, that's a primary consideration for HLA class 1 is what are those individual allelic effects? What peptides are they presenting? And does that result in a strong immune response or does it act as a decoy? A second characteristic is these differing expression levels that we think are modifying HLA allelic effects in the acquired immune response, which I showed you, but probably also the innate immune response, and we really haven't gone down that road just yet. HLA interacts with other variable loci differentially, and an example of that was with the microRNA gene. And this one, which I think really needs some concentration, is the variation within these HLA genes or nearby these genes are trans eQTLs for unlinked genes where we never knew before they had any kind of interaction with HLA whatsoever. We thought their functions were totally unrelated. And then finally, this pretty major role of class 1 in binding these innate immune receptors, the LILR molecules and the KIR molecules. LILR expressed on various types of cells, some on natural killer cells, some on dendritic cells, and they bind class 1, and that affects the activity of those cells. And I mentioned the effect of KIR in combination with B57, one of the ligands for KIR3DL1. So I'm going to stop there, and these are individuals. I've tried to mention the people as I went along. I'll just say this is my lab right here that is supported by the National Cancer Institute. But I actually have 6 people, these right here, that are supported either by the Gates Foundation or the Reagan Institute, and we're really lucky to have that kind of support. And I'll just mention the Office of AIDS Research, who supported the genome-wide sequencing of those B57 individuals. I'll stop there and be happy to take questions. Thanks very much for your attention.
Thanks, Mary. I think I'll start with a question. I'm very curious about the existence of high-frequency variants at unlinked loci that affect one another in the same population. And is that some funny form of selection, or is that human migration and admixtures just brought them together? Or?
Yeah. Well, we don't really know. So the Danny Dueck, I've spoken with him about this, and Danny thinks it could have to do with selection early on, maternal-fetal interactions. The thing about, so you're talking about the TIM3, for example. So the one thing I think about the TIM3 association is that, and the thing I kind of like, is that maybe during T-cell maturation, when the T-cell is in the thymus, there's a learning process, or somewhere along the maturation of a T-cell, that there'd be a learning process. That if you have a high expression allele, those alleles may result in more CTL activity, a stronger response, okay? And strong responses are bad in some cases, like autoimmune diseases. So upregulating exhaustion markers, for example, like TIM3, might be one way of controlling over-responsiveness of a given HLA-A allele. And it's a little hard to approach to know whether that's the case, but the thing is, we really have no idea why this is going on and how it's going on. But if you silence HLA-A, you can decrease the level of TIM3 after treatment, after stimulation. So we do think that's actually the decreased A expression is resulting in TIM3 change, not the other way, and not some global effect. Yeah.
So just follow. Do you see association for TIM1 and TIM4, which are also in the same chromosome 5?
Yeah, well, we haven't looked there. And we've only looked at TIM3, and this is really, we really aren't very far into this. We only looked at TIM3 because Nico had seen this association in the database. So we haven't looked at any other markers beyond TIM3. We've got a lot to do in that regard. Yeah. Because I'll tell you a lot. I can tell you one thing for sure is there is a lot of stuff in these eQTL databases that's not real. And I really feel strongly that if you want to look at an eQTL effect, it's not just using these chips, which is what's done here, to look at expression levels. Sometimes the probes actually, and they try to take care of this, but sometimes the probes can cross-react with other genes, so you may not be looking at the gene that you think you are. So you have to go back and do qPCR in order to know whether those are real. And the one with TIM3 we think is real.
Hi, great talk, thank you. Do you know if any of these genes are under positive selective pressure?
Oh, yeah, like, okay, so the class 1 and the class 2 genes are under strong balancing selection, okay? As I was saying, there's support for heterozygote advantage in viral disease. And in general, the way we think about it is the more heterozygous you are, the greater the repertoire of peptides you can present. So there are good things about being heterozygous. And if you actually, the evolutionary people who have actually done those tests, there is selection for, there's balancing selection at the HLA class 1 and class 2 genes. Absolutely.
And among the other genes that you found?
So here, you know, there's selection, okay, in KIR3DL1, Peter Parham's group has shown that there are, if you do phylogenetic tree, there are 3 clear clades, and they've shown that those clades are under selection pressure, okay, to remain across populations. So every population they looked at had those 3 clades, even though the alleles within those clades might be different. So there is selection pressure to maintain those. I don't know of anyone that's shown actually balancing selection for KIR3DL1 alleles, yeah.
Hi, Mary. That was really cool. Is there any information about susceptibility to infection of people with Crohn's disease? Or progression to AIDS?
Yeah. So we kind of, I'm part of this IBD consortium, and you know, I brought this up before, but there is no cohort that we know of. I don't know of any HIV-infected patient who has Crohn's disease. And we certainly don't, there could be, and there probably are, but they're probably really rare. And you would never get enough samples to be able to do a decent genetic analysis. But maybe, but who knows? I don't know. The idea about thinking about these diseases, I think in some context, maybe at some point, we'd be able to look at pairs of diseases where a given HLA type has alternative effects. Like, for example, B57 is protective against HIV, but it's thought to be associated with psoriasis. So are people who have B57 are controllers more likely to have psoriasis? I don't know the answer.
And just, it's not quite an underwear experiment, but weren't there experiments in mice?
Yeah, and I should have mentioned that. So subsequent to Bernard buying all that underwear, there actually were, and you know, these experiments were slightly more elegant, where you know, they looked at inbred strains of mice, and the one inbred strain definitely preferred to mate with mice of a different inbred strain, and that I think was, you know, pretty nicely shown. And the idea there was that, and it had to do with mouse urine, that H2 is actually found, H2 molecule, class 1 molecules are actually found in mouse urine, and there was some idea, but I don't know, you know, how much of this is real, is that depending on MHC type, there would be a different scent of the urine. And I don't know how valid that is, but that was sort of the idea, yeah.
Could I add to that? I've heard a story that in order to follow it up, they got perfume makers to smell the urine. And one
Was that a Chanel commercial? Yeah.
Could you please comment on why it is so difficult for a diverse virus like HIV to make a mutation within that epitopes that are identified by HLA and easily escape? So when you say these are protective HLA, what's the mechanism?
Yeah. So it's thought for B57 that B57 targets a lot of GAG epitopes, and the general thought is that the more conserved an epitope where if it was did undergo an escape mutation, that it would affect the fitness of the virus. So it's really hard to actually show that perfectly, and I think one thing I think is that it's really hard to pinpoint why. There are so many things about these molecules, which is what I've tried to show, that could affect how effective they are against a given virus. For sure, HIV undergoes mutations that associate with HLA, that are driven by HLA, and some of these protective alleles may be because they're hitting the virus at a place that the virus needs that in order to remain fit. It needs to maintain its sequence in order to remain fit. So, but I think it's probably a simplification, actually. I think probably there are a lot of things about these molecules that and so trying to pinpoint it is going to be difficult.
Is there any connection with drug resistance? Some of these peptides may be different in case of drug resistance. Are they selected differently? Anyone has advantage or disadvantage?
Yeah, so that's a good question, and I don't know of work where somebody's really tried to approach that. At one point we tried to work with Doug Richman on looking at effect of HLA in people treated for HIV. But it was a problem because the treatments were so diverse, and maybe they're not as diverse at this time. We could never really get enough people it was a huge variable for us, so we really couldn't approach it in a way that we felt very secure. But it's a good idea, and I think if we had enough people that were treated. And I think another problem with that was that it wasn't so clear that all the people were staying on their meds. This was at a time when a lot of the meds were really difficult to take. So there was a compliance issue in there too that we couldn't really address. But maybe now that's something that we need to revisit.
One perspective of looking at Carlson's work would be that if you could map all the epitopes that are being recognized by an individual, you can map it back to the the HIV proteome and pick out epitopes that are being masked. And you could ask questions like, why are they being protected from immunogenicity? Do you have any comments on that?
I'm not sure so in those individuals, we do know which epitopes are restricted, which HIV epitopes are restricted by each HLA type. And so we have that information. So I'm not sure I understand the approach that you're saying, is that now that we know that information, we know which epitopes
You could map it back to the HIV proteome to find out what other peptides are not being represented in the epitope section.
Yeah.
And see if there is a functional cause for why it's happening.
Right. Like that maybe some alleles are attacking peptides that are going to that if they mutate there, it affects the fitness of the virus. Is that what you're saying?
Yep.
Yeah. It's a little difficult because and we do have that information, and I think Philip Goulder has done a fair bit of that work, but first of all, there aren't single peptides that are recognized, so we need to fish through which one is consistently, to which one are they responding renders consistent protection. And we get into power issues there as well. So if you start thinking, and not only that, you have to think about HLA as a whole. So we can't just look at HLA-C, we have to look at B and A. Now you're talking 6 different HLA molecules, and you have to start thinking about all the epitopes. So it gets really sticky to try to unravel. But as I said, there's a real body of literature where having more CTL responses against GAG epitopes is conferring protection, and it's based on that kind of data that was generated by Toombi and Philip.
What happened to the box of underwear? Does the government have it somewhere?
Well, no, when I left Duke, I checked this before I left, it was still there in his office. And so it'd be really curious to know where that is now.
I will take
They're old type of underwear. They're probably an old version that wouldn't be comfortable anymore.
I don't need any. We'll take 2 more questions and then we'll go to the poster session.
So you mentioned the importance of different expression levels of the HLAs. And maybe a naive question, but is it actually known why they are expressed at different levels in different people?
Yeah. So one of the reasons for differential expression is that microRNA binding site. And, but that does not explain, like, I think one of our highest alleles actually has the microRNA binding site, and still it's high expression. So we've begun to look in the promoter region with Steve Anderson, and we're trying to figure that out. For HLA-A, Veron has done a lot of work looking at methylation. Methylation doesn't seem to have any effect on B or C expression, but it does have, there's a nice correlation with the level of methylation and A expression, where the more methylated, the lower expression it is. So some of those things are beginning to be teased out, but the thing that is just so wonderful is that even though A, B, and C have this common function of antigen presentation, those 3 genes are so different from one another in many, many ways, and that's one way. B is totally flat. B alleles are all expressed at the same level. People don't really vary much in their B expression level. So really quite different characteristics of those loci.
One more question.
Yeah, I'm wondering whether you have any evidence that for mother-child transmission of HIV, epigenetics might play a role to confer partial resistance or vulnerability to HIV infection.
Okay, well, one thing that's clear-cut, and I think it's clear-cut at this point because it's been shown in several studies, is that the more sharing, okay, so the mother and the child, of course, are going to share on one haplotype. And, but if the father has a haplotype that's similar to the mother, and the baby inherits, the more the baby looks like the mother at HLA, the more likely that baby is to become infected. And so now, you know, that's not as big of a problem because the babies, you know, the mothers are treated before the baby's born. But I think it's pretty clear, that finding is pretty clear. The more sharing of HLA, the more likely a baby will become infected. And I don't know if Jalen Gappa is here yet, but is he coming? Oh, yeah. But anyway, he could comment on that more. But yeah, definitely. So in that regard, I don't know if that's really addressed your question or not, but that seems to be the case. I can tell you, pretty much, we don't think, and I know some people don't agree with us, but we don't think that HLA and or cure protect people from infection. And we feel strongly about that, though there are reports out there that some do. I don't think HLA and or cure variation protects from HIV infection, except for that case with the baby sharing with the mother, and I think that's a different mechanism. Yeah, I think that could be transfer of cells. I don't know.
So thank you very much.
Thanks a lot. Thanks a lot.